Okay, this is the next part. This is number 3, looking at the outcomes of complement activation. We're on slide 14. So the first outcome is opsonization, and opsonization simply means that those molecules, in particular C3b, stuck to the outside of our bacteria, make it easier for a phagocyte to recognize, stick to, and engulf that bacteria. So we have in our diagram on slide 14 our little red bacterium covered in our green circular C3bs. On our macrophage we have receptors for C3b. Our bacteria covered in C3b can dock, or indeed our macrophage can dock, and then our macrophage can engulf and eventually destroy our bacteria. So macrophages, very important in this mechanism. Neutrophils are very important too. Opsonization, making it easier for phagocytes to engulf and therefore destroy bacteria. Next on our list, slide 15, is inflammation. Number 2, inflammation. So the outcome of complement activation is also inflammation. And again, we can have all of these outcomes happening at the same time. We might have phagocytes eating up opsonized bacteria, and in addition to that we have all of the stuff of phagocytosis happening, and then in addition to that we have membrane attack complexes forming, as we'll see later on what a membrane attack complex is. And inflammation in general is always the end result of any pathway in the immune system. Not just the innate immune system, but the adaptive immune system. At some point, a microbe gets into your body, some system identifies and mounts a response, and through various mechanisms, inflammation occurs. So in this case, we have our complement proteins getting split. We have C5 getting split into C5a. And C5a is what's called a chemoattractant, in that it attracts phagocytes to the area of infection. C3a and C5a, they increase the permeability of blood vessels. So our blood vessels are lined with these cells, very thin cells that make up the walls of our blood vessels. And these very thin cells that make up our blood vessels are called endothelial cells. These blood vessels line our capillaries, and it's through the little gaps of these cells that all of that fluid is lost out of the blood. And our red blood cells flowing through our capillaries, all that fluid is lost into the tissues. When C3a and C5a get together, they make these blood vessels much more leaky, and that means a lot more fluid is getting lost into the tissues. So more interstitial fluid is lost, and we'll look later on at a result of that is the swelling we see in tissues. So you get a splinter, for example, in your finger or your thumb, and it gets red and swollen. The swelling is because there's a lot more fluid being lost out of your blood vessels into your tissues. But as that additional fluid is being lost, it also allows cells like neutrophils to squeeze through the gaps and get into the tissues and start destroying bacteria. And it allows things like monocytes to squeeze into the tissues and become macrophages. Neutrophils and macrophages, two very important phagocytes, cells that eat. These complement pieces, C5a and C3a, C5a, also cause mast cells to release their granules. So mast cells are very similar to basophils. They are often termed with the granulocytes. Mast cells just kind of hang around in tissues, and they kind of a whole bunch of chemicals waiting around for activation. When they get triggered, they release a bunch of chemicals, including chemicals like histamine, that we're probably very familiar with in terms of inflammation. And that histamine then works its way into the body with a whole lot of other inflammatory chemicals and causes all of the outcomes of inflammation. So on slide 16, we have the pictures from the book of the inflammatory response. We have a splinter getting into your skin, penetrating your defenses, bringing with it a whole bunch of bacteria. In this picture, we have a macrophage just hanging out in the tissues, and that macrophage will use its toll-like receptors to identify bacteria, engulf them, and that macrophage itself will release a whole bunch of cytokines. But also in our tissues, in the fluid, are our complement proteins, and they are also detecting the presence of these invading bacteria, and they are triggering that activation cascade. All of the pieces, the C3 being split into C3b and C3a, C5 being split into C5a and C5b, and in particular C5a, C3a, important in attracting more phagocytes to the site of infection. We have the recruitment of neutrophils and more monocytes becoming macrophages. As the monocytes and neutrophils arrive at the site of infection, the increased leakiness of the blood vessels allows them to squeeze out and get into the tissues and start to destroy those bacteria. Slide 17. The third outcome of complement activation, lysis of foreign cells. So once a bacteria, say, has been targeted for destruction, a whole host of complement proteins will attack, and they will lodge themselves into the membrane of a bacteria, in particular into a gram-negative bacteria. We'll talk about gram-positives and gram-negatives later on. So our gram-negative bacteria in our tissues, we have the attack from all of these complement proteins, and that attack puts a little hole in the membrane, and all of the cell contents leak out, and the cell is killed. So the three outcomes of complement activation, they help with this process called opsonization, which means that when the phagocytes get to the site, all of the nasties have been targeted. The phagocytes know what to go after, and they have an easy time picking off those targeted, let's say in this case, bacteria. Then we have inflammatory response. We have the attraction of phagocytes to the site of infection. We have the increased leakiness of our blood vessels, bringing more fluid into the tissues, but also allowing those phagocytes to leave the blood and enter the tissues. And then we have the lysis of foreign cells, the formation of membrane attack complexes, MACs, as all of those complement proteins insert themselves into the membranes of, in particular, gram-negative bacteria, puncturing the membrane and allowing the cell stuff to leak out. All of these outcomes can happen at the same time. All of them can be triggered by all the different ways that our complement system was activated. So I mentioned before that C3b likes to stick to all kinds of cells, including our own cells. But our own cells don't trigger any of these outcomes of inflammation. If they did, we would start to attack our own tissues. And indeed, there are some diseases that are termed autoimmune diseases, where we do, in fact, attack our own tissues. But generally, we don't attack our own tissues because our own cells have these regulatory molecules. You can see there on a host cell, the C3b sticks, but its activity is blocked by the regulatory protein. Okay, so I'll stop this section right there.